All right, so guys, a little bit of genetics recap in case you aren't quite understanding. We're talking about living things and why living things, why organisms look the way they do. Okay, and we know it comes from the DNA, we know it's in the nucleus, and we know we get our DNA from our parents. So an organism is basically a collection of traits. Okay, traits meaning how we look, how our body functions. Okay, and we get these traits from our parents. Okay, the word heredity means the passing on of traits from parents to offspring. All right, so if mom has brown eyes and dad has brown eyes, baby most likely will get brown eyes. Okay, maybe green, maybe hazel, but this right here, the passing on traits, would be heredity. Okay, now these traits are stored on our chromosomes, and there are codes on our chromosomes in our genes. Okay, so the genes has the code that will make you look the way you do. And that's what we're going to be looking at for the next couple of days, the next few slides. All right, so let's see what's next. Each trait that we have in our body, whether it be the way we look, how tall we are, how our body breaks down proteins, can come in different forms. Okay, so we know there are different eye colors. We know there are different hair colors. We know there are different heights. Okay, and all of these things come in different forms, and we call them an allele. Okay, so the different form of a trait is an allele. Now, for example, your hairline. Either you have a straight hairline, like this woman right here, or you have something called a widow's peak. So that is one possible allele, hairline and the different forms. Okay, dimples. We can have dimples or no dimples. That is another form of a trait, an allele. And like we said before, brown eyes versus blue eyes, again, both examples of alleles, different forms of a trait. Now, the scientific study of how all of this gets passed on and why we look the way we do is called genetics. Right, so genetics started back in the day with this man called Gregor Mendel. Okay, here's a picture of him right over here. Gregor Mendel was an Austrian monk. I believe he had a science background, but in his monastery, he spent a lot of time in the garden. He was a gardener, and specifically, he studied pea plants. Okay, and by looking at his pea plants, he started to realize that certain traits got passed on over and over again. Okay, and he wanted to figure out why these traits got passed on the way they did. Okay, at this point, they thought that kids were a mixture of their mom and dad. Okay, but when Gregor Mendel started to do his research, he saw that it wasn't always an exact mixture. All right, so he studied his pea plants, he made scientific observations, and he looked at how traits were passed on from one generation to the next. Now, Mendel's research never really got big when Mendel was alive. Okay, people didn't really accept what he was saying. Until years later, a lot of other scientists came to the same results. This occurred after Mendel died, but since then, he has been known as the father of genetics, the first person to really realize how genetics works. And right now, we are going to look at what Mendel saw. All right, so Mendel took two pea plants. He took a round pea plant, and he took a wrinkled pea plant. All right, now we a lot of times think of wrinkles as when you get old, but certain peas are wrinkled, they're born with them, and other peas are perfectly round. So he took these peas and he mated them with each other, which we call a cross. All right, so for plants, it involves pollen and the stamen. When Mendel did this, he wanted to see what trait got passed on. He wanted to see what the babies were going to look like. And every single time he did this, every baby was round. Right now, this kind of surprised Mendel because one of the parents was wrinkled. What happened to the wrinkled trait? He never, ever, ever saw a wrinkled baby. They were all round. Okay? And we call this baby a hybrid because it was a mixture of the round and the wrinkled pea plants. It got genes from both parents. All right, so Mendel was curious. Why was it always round and never wrinkled? Here's what he was able to infer. Okay? He was able to figure out that the round trait must have been dominant. Okay? What this means is that this trait will take over. Okay? Now, if this trait takes over, it's the one that shows up, it dominates, that means the other one gets hidden. Okay? The wrinkle trait, he said, was recessive. Okay, so the P, the baby P that came out round, had the wrinkle trait in it, but the round trait was dominant and took over, making the baby look round. Okay. Now, when Mendel was doing this, a way to help him explain it, he assigned letters to the dominant and the recessive trait. So he used a capital letter to represent the dominant trait because the capital letter takes over, okay, and a lowercase letter, little r, to represent the recessive trait. And you can really use any letter at all that you want. Okay, so now we're going to do something called a Punnett square to help show Mendel's results, to show what he saw. So a Punnett square just helps us to predict what our kids are going to look like. So we're going to go back. We're going to look at his P's. Okay, if we look right down here at this diagram, it is showing us that we have our round, let's call it round instead, and we have our wrinkled P's. Okay, now these letters are representing the genes. So the round P'd mom, let's say, has two big S's. The S's, capital S's, represent the dominant traits. And there's two of them because every organism has two copies of each chromosome. One came from mom and one came from dad. The wrinkled P has the recessive trait. Okay, so we have two little s's for our wrinkled P. Okay, same idea. One comes from mom, one comes from dad. So now we're going to do a Punnett square. And here's how we do it. It's actually pretty simple. You make a box, kind of looks like a window. Okay, and we take one parent's genes and we put it on top of the box. So big S, representing the round parents. Okay, now we're going to take the other parent's genes and put them on the side, little s, little s. Now that we have it set up, all we do is bring our letters together. Okay, so now all we do is bring our letters together. Coming down here, we have a big S, and coming in this way, a little s. Okay, moving over, bringing it down and across, that gives us a big S and a little s. Again, bringing it down and across, big S from the top, little s from the side, and again, bringing it down and across, big S, little s. So if we look at our babies, every single time we have the combination of big S, little s. Now, since the big S is the dominant trait, even though we have the little s, the dominant trait takes over. So no matter what happens, all of the babies with this big S are going to come out round. It carries the gene for the wrinkle trait, it's just not showing up. Now, when we are talking about these Punnett squares, we have two new vocab words. Okay, genotype, these are the genes, okay, your genetic makeup, and these were the letters that Mendel used. Okay, so either big S, big S, or little s. These are all possible genotypes. The phenotype is the physical appearance, okay? It's the way the organism looks. So if we have big S, little s, since there's that big S, the dominant trait will take over. Again, our two big S's also will show the dominant trait, but two little s's will show the recessive. Okay, so our genes affect the way we look. Our genotype affects our phenotype. Okay, and here it's showing our genes on the chromosomes, coding for how the organism looks. Now, the letters that we were using, again, just represent the genes. There's not actual letters on your chromosomes, just a way to represent it. Skipping this for right now, skipping this for right now. Okay, so for example, we all know that we have two pairs of each chromosome. One comes from mom, one comes from dad. Okay, again, one from mom, one from dad. So for each chromosome we have a matching pair. Okay, 23 matching pairs to be exact, giving us 46 chromosomes. Okay. So mom could give us a dominant trait. Mom gives us a big S, our dominant trait, and dad gives us a recessive trait. Okay. Now, we have the same gene, but the different types of alleles. Okay. In this case, in our yellow gene down here, maybe we have, using T's right now, it doesn't matter what letter you use, maybe we have the same type. So both the big T's, the dominant trait. Okay, and down here, we will have little f's. Okay, again, doesn't matter what letter you use. Now, the fact that these two genes, our two S's, are different, one is dominant and one is recessive, we call that heterozygous. Okay, hetero means different or opposite. So heterozygous is describing that we have two different alleles. Okay, and whatever letter we use, it doesn't matter, but one is dominant and one is recessive. Okay, on the other end of it, if we have two of the same, like our F's down here, they are both recessive, that would be homozygous. And we know homozygous means that the alleles are the same. Okay, so we could either have a homozygous recessive, or we can have a homozygous dominant. Okay? So let's see some examples right now. We have our Punnett square, and it's telling us we want to cross a homozygous dominant with a homozygous recessive. Okay, homozygous means the same, and dominant means it's a capital letter. So pick whatever letter you want. Let's go with A. That means we're going to have two big A's. Okay, again, we have homozygous, but this time it's recessive. So we're going to have two of the same little A's. And then you just set up your Punnett square. Big A, this is one parent here. Little a is our other parent. It doesn't matter where you put them. And just bring your boxes together. Okay, so we get big A from the top, little a from the side, big A from the top, little a from the side. Again, big A from the top, little a from the side, big A from the top, little a from the side. Okay, so when we cross these parents, every single one of our babies is big A, little a, which means they are all heterozygous. Okay, they both have the different traits. All right, let's see. Any more examples right now? There we go. Heterozygous versus homozygous recessive. Okay, so heterozygous means the traits are different. Let's go with R's. Big R, little r. Okay, homozygous recessive means they're the same, but they're little, both recessive. Okay, so we're going to put our parents, little r, big R, little r. Bring your boxes together. Big from the side, little from the top. Big from the side, little from the top. Little, 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 little. Okay, so now we're seeing some variation. Both of these kids on the top are going to be heterozygous. Okay, and on the bottom, we have homozygous recessive. Right, and our last example, heterozygous and homozygous dominant. All right, so let's go with T's. Heterozygous, they will be different, big T, little t. Homozygous dominant, both big T's. Okay, put our parents down, big T, big T, little t. do your Punnett square, 2 big T's, 2 big T's, big little, okay? So this time, again, we're seeing differences, but each child is going to show the dominant trait, because the dominant trait always takes over. All right, I think that's going to be it for right now. We will come up with another video to show the second half of the note packet.